As probably most of you know, basal cell carcinoma is the most frequent cancer of all forms and is increasing in most countries, also in Sweden. We have an interest in basal cell carcinoma due to its impact on medical care and the amount of cancers every year.
We started by collecting biopsies from the patients, and we took one biopsy from the central part of the basal cell carcinoma, and then we took one biopsy from the patient's own healthy skin. From these tissues we extracted RNA, and the plan was to run a microarray on these samples. We managed to get some good samples for the analysis because microarray is a very sensitive analysis method. In each sample you can measure up to 50,000 gene expressions. So by doing that we compared a group of tumors with their normal tissues. And when analyzing the data we used strict correction methods, and we chose the differently expressed genes that had a fold change of more than 5. And by doing this we believe that we might have a higher biological relevance in the genes that were filtered out through the analysis. As we are interested in metabolic activities in the tumors, because that seems to be very important for cancer cell turnover, we selected genes that have an involvement in amino acid transport and metabolism. So from the 4 genes, 3 of them are members of the solute carrier family 7, and one is an enzyme that degrades the essential amino acid tryptophan. And just so knowing that these genes are actually upregulated in the tumors compared to normal tissue, we did individual analysis of each tumor and compared it to every patient's own control skin. In the protein analysis, we saw that 2 of the SLC7 members, A5 and A8, is actually expressed by the tumor cells. TDO2 is expressed, as it seems, by the immune cells and is also found in the stroma of the tumors. For SLC7A7, we could locate it to the stratum granulosum of the epidermis, and by being found there, it has probably little to do with carcinogenesis. However, we could see that higher protein expression of A5 and A8 also was correlated to high gene expression and could have actually an influence on the carcinogenesis, being the transporting mechanisms of essential amino acids and possibly having a role in cellular metabolism. We have found new candidate proteins that are potential targets for inhibitor drugs, and that needs to be further elucidated in future studies.